Welcome to the video on DNA replication. So let's think about this. When you're first made as a baby, you get one set of chromosomes from your dad and one set of chromosomes from your mom that combine to make a single cell. So you start life out as a single cell, but I don't think you're made of just one cell now. You have tons of cells in your body, and they all have the same DNA that was in that original cell that made you as a baby. So, how do we get that DNA into every single one of our cells? Well, DNA has to copy itself, and it does that through a process that we call replication. So, there's two major things that help DNA copy itself. Number one was Chargaff's rule, that all A's bond with T's and all C's bond with G's. And the other is that there's two strands of DNA. Basically, what's going to be happening with replication, think of splitting the DNA strands apart so that there's no bases paired with each other. You only have an A over here and a C over here. There's no A and T, C and G base paired. And then you take those DNA strands and you put them in a soup of a bunch of free-floating nucleotides, so a sugar, a phosphate, and an A base, a sugar, a phosphate, and a T base. Well, they're going to want to connect with each other. They're going to want to form those base pairs. That's essentially the idea behind what we're going to be doing. You start out with one strand of DNA, it splits itself open, it copies itself, and you get two new strands of DNA. So why do we have two copies of all of our chromosomes, and where do we get each set of chromosomes from? And what are two characteristics of DNA that make it possible for DNA to copy itself? We're going to focus on three main steps in replication. Number one, you have to separate the strands. And we do that using an enzyme called DNA helicase. So DNA helicase unzips DNA. Think about it as a big zipper. And it does that by essentially breaking the bonds between the base pairs. So if you have an A and a T bonded together, helicase will break them apart. Second, we have to make our new strands. And we do that using an enzyme called DNA polymerase. So now we have two separate strands of DNA because DNA helicase separated them. And we're going to have DNA polymerase come in and add free nucleotides to that strand by matching the bases, A with T and C with G. The only problem with DNA polymerase is that it only works in one direction. Remember that DNA was directional, anti-parallel. One strand goes in one direction, the other strand goes in the other direction. So, how does that work? What ends up happening because DNA polymerase can only go in one direction? What ends up happening is you have one strand that's replicated continuously, or non-stop, and another one that's replicated in short little bursts. We call the one that replicates continuously the leading strand, and the one that replicates discontinuously, or stopping, the lagging strand. Think of it as lagging behind. So in this case, we're going to have DNA polymerase copying from the 5' end here to the 3' end. That's how it adds the nucleotides. And as DNA helicase unzips in this direction, it can just keep going and keep copying. But in this direction, it can't do that. DNA polymerase is going to want to move from 5' to 3' in the opposite direction, but it can't do that until more DNA is unzipped. So until DNA helicase opens up more of the DNA to copy, it's going to only be able to copy it in short little bursts. So we end up calling those Okazaki fragments after the man who discovered them. Now what ends up happening is we have a replication bubble. Rather than just the fork, you're going to actually have a bubble to help replication go faster. And we can do it in two different directions, but one of those directions is going to have those Okazaki fragments. Why do you have to separate the DNA strands? What are Okazaki fragments and why do they exist? The last part is we have to attach those fragments now. So you have all these Okazaki fragments, but they make up a discontinuous strand. The strand isn't brought together. And that's where DNA ligase, our third enzyme, comes in. And it attaches these Okazaki fragments by binding the phosphate sugar backbones of DNA. So what are the three main enzymes needed in DNA replication? How many DNA strands are made from the original strand? So first, we talked about DNA helicase separating the strands. Second, DNA polymerase makes the new strand. And finally, DNA ligase attaches the fragments. That's all that we're going to talk about for DNA replication. The whole process of replication is what we call semi-conservative. And what we mean by that is that there's a little bit of the old DNA molecule in every new molecule created. Think of it this way. You have the pink parent strand here. It's going to unzip, and half of it's going to go this direction, the other half is going to go this direction. And those are copied. So in the end, you get one strand that's new and one strand that's old to make your double helix. So that's semi-conservative. We're conserving or keeping part of the old molecule. And our last questions are, does any of the old DNA molecule move on to the new DNA molecule? And why do we have to replicate DNA? We didn't talk about that one in the video, but I want you to think about it, get an idea, and come to class ready to talk about that tomorrow.